Hello, I'm Peter Gibson. I'm a respiratory physician, and a lot of my time is spent trying to diagnose the cause of respiratory tract infections. Sputum is an excellent sample to make the diagnosis of respiratory infections. It has two main points in its favour. Firstly, it is a good source of lower respiratory material, which is where the infection is, and secondly, this material can be obtained non-invasively with minimum discomfort to the patient. In this video, we'll see techniques to ensure the best success rate for obtaining good sputum samples. To get a good sputum sample, you need to pay attention to three key details. Firstly, have the patient cough. Secondly, clear their throat. And thirdly, empty their oral contents and spit into the collection jar. Each of these three techniques is crucial to getting an adequate sample to give the best success rate in diagnosing respiratory infections.
Pre-bronchodilator spirometry.
Hello, I'm Michael.
Hi, I'm Kevin.
Hi. Today we're going to use this equipment to test your lung function, to see how efficient your lung filling and then empty. Basically, we need to do the 3 times to pick up the 3 best results, at least 3 times. And have you withheld your asthma medication in the last 6 and 12 hours?
Yes, I've withheld my Ventolin for more than 6 hours,
Yep.
and I've withheld my Seretide for more than 12 hours.
That's good, okay.
To perform spirometry, you will need the equipment shown that is also connected to a computer.
I need to set up the machine first. Once I set up the machine, and you can take this mouthpiece and put your mouthpiece into your mouth, make sure it's sealed properly, and then you put your nose clip onto your nose. So, and then you can start nice breathing out a few times to feel how you're comfortable with it. Once you are ready, you feel free to take a big, deep breathing to fill up your lung. As soon as it's full, you blast out as hard and fast, and then you keep on going, breathing out, completely empty your lung. Once the lungs are completely empty, and you start breathing, and I'll let you know to stop it. During the testing, you don't stop it without telling you, and don't take the mouthpiece and nose clip off. Right? And when you're breathing out, make sure you don't bend your body too hard. And make sure you sit nice straight, so because otherwise it affects your results. As I said, we need 3 valid results. Okay, so are you okay? You ready to go? Any
Yes.
questions?
No, I'm good to go.
Okay, let me set up the machine so you can put things on. Set the machine, put the nose piece, put mouthpiece in. Yeah, start normal breathing out. In, not to rush if you don't feel ready. But as soon as you're ready, take a big deep breathing and blast out as hard as possible and keep on going until you completely empty, and then you cycle back. Okay, good. Yeah. Okay, very good.
After performing spirometry, the computer will generate flow volume loops. The flow volume loops on screen show typical results for an asthma patient, both pre and post bronchodilator, in this case Ventolin, and a healthy control result. Administration of Ventolin. To give Ventolin to the patient, you will need a spacer and a Ventolin puffer.
Is we give you 4 puffs of medication, Ventolin called, and to retest your lung function to see how your lung responded to this medication. Have you used this spacer before?
No, I haven't.
Okay. You can do very nicely. One method is as soon as you press 1 puff and you have 3 times breathing out for 3 times. Ready to go? Make sure you empty your lung first. in, out, in, out. Okay.
Repeat this procedure with the patient 3 times for a total of 4 puffs of Ventolin. An alternative method of Ventolin administration is where the patient inhales Ventolin from the spacer, and instead of 4 breaths in and out, the patient holds their breath for 10 seconds before exhalation. This method is also performed for a total of 4 times. After Ventolin administration.
Alright. Now we wait for 10 minutes and then we retest your the lung, exactly the same last time, okay? To see how good your lung responded to these medications.
Post-bronchodilator spirometry. Again, you will need the equipment shown for spirometry.
Start normal breathing out. In, not to rush if you don't feel ready. But as soon as you're ready, take big deep breathing and blast out and hard and possible and keep on going, keep on going, keep on going, keep on going until you completely empty and then you cycle back. Okay, good.
Sputum induction. For the sputum induction, you will need the following nebulizer equipment. Fill the nebulizer cup with 4.5% saline to the recommended hashed level. Next, weigh the nebulizer cup with tubing attached and record the weight. For the sputum induction, you will need a fully set up nebulizer, hypertonic saline, and 2 sample collection jars. Ensure that you have set up the mouthpiece components correctly as shown in the picture.
To induce your sputum, we will get an analysis in the lab. Now, this equipment is that help you to, when you inhale this saline, it's saline in there, and will lose your sputum and get you to produce the sputum. Now we're going to do about 6 different times. The first is 30 seconds, then 1 minute, and 2 minutes, and 3 lots of 4 minutes. Now, during the inhale these saline, you have to make sure you seal the mouth properly. And if you can't hold it anymore, you just raise your hand and we can stop. And each time we get the saline, we will retest your lung function. Okay. So, before we do this, we also need to rinse your mouth 3 times to make sure your mouth. Yeah. And any questions?
No.
Okay. All right. So now, yeah. You rinse your mouth and spit it into the jar. Okay. Third arm. Great. Okay. Now we have those 2 jars to collect your sputum. This is the first one. If you have a sputum, you feel free to spit into this jar. If you don't have it, now we have another one for later on after you inhale the saline. Now, to produce your sputum, we need to follow the 3 steps. First is cough up, and second is hack, and third one spit into the jar. So let me demonstrate this. First, and then spit it into the jar. Okay? Alright. So are you ready to go? Do you have any sputum now?
No, not at the moment.
Okay. Alright. So I gotta set up time and you start to put the things onto.
Okay. So nose clear.
Nose clear? Yeah. And make sure you seal your mouth properly.
Yep.
Yep. And just normal breathing out. 10 seconds. Okay, stop. Now, in about 1 minute time, we will retest your lung function. So how do you feel now?
I think I've got a little bit of sputum to come
Okay, so if you got it, so feel free to produce it. Good.
Wait for 1 minute before retesting the patient's lung function.
It's almost 1 minute now, so we are going to test your lung function. Exactly the same as last time, but this time when you breathe it out, it will be shorter. But you don't stop it, I will tell you when to stop.
Okay.
Okay. All you need to keep exactly the same effort like last time. Okay?
Right, so nose clip on.
Let's clip on. Okay. Alright. So just not to hurry, normal breathing. Yep, sits properly. Yep, normal breathing. Out, in, out. When you're ready, you take a big, deeper breathing and pass out. Yep. Okay, stop. Yep. Take the nose clip off. Very good. Very good. Okay. Let me calculate your lung function. Your lung function is within 15% of baseline, so we can continue the sputum induction.
If the patient's lung function continues to be stable within 15% of baseline, continue the sputum induction for a 1 minute, 2 minute, and 3-4 minute intervals, retesting the lung function after each interval. Alternatively, if the patient lung function has fallen below 15% of baseline after saline induction.
So let me calculate it. So looks like your lung function has dropped more than 15% from baseline. So what are we going to do? We're going to have give you the Ventolin again. We give you 2 puffs, and then we retest your lung functions to see whether you recovered.
Okay.
And if you recover, we can continue. If not recover, we give you another 2 puffs. Okay. So you same as last time, and when I press puff, and you start 4 times breathing out, breathing out. So
Okay.
I'll give you 2 lots.
Yep.
Okay.
So empty my lungs.
Yeah, empty your lung first. Okay. Yeah. In, out. In, out. In, out. In, out. Okay.
Repeat the Ventolin administration 1 more time for a total of 2 puffs given to the patient.
Alright, so we're going to wait for 10 minutes and then we retest you.
Okay.
Okay.
If patient lung function recovers, continue the sputum induction. However, if patient lung function does not recover, give 2 more puffs of Ventolin using the same method just shown. Always refer to the sputum induction flowchart for the correct procedure. Here is the sputum induction flowchart. If the patient lung function does not recover after 2 Ventolin administrations, see your supervisor. Again, always refer to the sputum induction flowchart. When the sputum induction is finished, weigh the nebulizer cup with tubes attached on the scales and record the weight. Saline nebulization rate calculation. To calculate the nebulization rate, subtract the post-nebulizer cup weight from the pre-nebulizer cup weight and divide this by the total time in minutes the patient has spent on the nebulizer. If a full saline induction has occurred, this will equal 15.5 minutes in total. The final answer will be grams of saline aspirated per minute. Nebulization rate in adults should be greater than 1 gram per minute. Anything lower can indicate technical problems. Organise for the laboratory technical officer to collect the sputum samples. This may include saline-induced sputum and spontaneous sputum. Samples should be transported in a biological specimen carrier bag. Now we will go to the laboratory to process the sputum samples. Biohazard safety cabinet cleaning. Spray 70% ethanol onto the work surface and wipe entire surface with paper towel to clean. Sputolysin DTT dilution. To dilute the sputolysin, you will need the following equipment. Label a clean 15 ml Falcon tube with DTT, your initials and the date. Nitrile gloves are worn for this step as they are more protective against needle stick injury. Take out a 1 ml syringe and a 25 gauge needle and firmly attach them together. Very carefully remove the needle cover, ensuring to pull the cover away from yourself to avoid injury. Pierce the needle through the rubber stopper of the sputolysin bottle. Ensure that all bubbles are removed from the syringe before drawing out 1 ml of sputolysin from the vial. Dispense the 1 ml of sputolysin into the DTT labelled 15 ml tube. Discard both the needle and the syringe into the sharps bin. Add 9 ml of distilled water to the 1 ml of sputolysin in the labelled 15 ml Falcon tube. Diluted sputolysin, or DTT, expires after 48 hours at room temperature. Mix the sputolysin and water together by inverting the tube. Identifying saline-induced sputum plugs. To separate the sputum plugs from the saline-induced sputum sample, you will need to have the following items shown. Ensure they have been cleaned with ethanol before placing inside the biohazard safety cabinet. Add the patient details and total sputum volume to the sputum processing worksheet. Invert the saline-induced sputum sample onto an open petri dish. If the saline-induced sputum sample is of good quality, then you will not need the spontaneous sputum sample. This is at the laboratory processor's discretion. While the sample is running out of the jar, get your autoclaved forceps and scissors from the autoclave bag. You may need to pull the remaining sample out of the jar with the forceps. Ensure that the majority of the sample is taken out of the specimen jar. Next, you will need to record the macroscopic details of the sputum sample. Use the Bronco test for grading sputum purulence as a color guide. Estimating the number of sputum plugs. The black plate underneath sputum will help you to identify sputum plugs and provides the size of one plug to scale. These images show several sputum samples and indicate the color and number of plugs in each. Selecting sputum plugs from saliva. Next, you will select the sputum plugs, removing them from the saliva and place them onto the petri dish lid as shown. Initially, you will need to identify sputum plugs in the sputum sample, and then, holding the plugs with forceps, pull them out of the sample and cut off any excess saliva. When you have finished selecting plugs, place the scissors and forceps into 70% ethanol to clean. Sputum you have selected should consist of mostly sputum plugs. Dispensing sputum plugs. To accurately measure the volume of sputum plugs, you will need a positive displacement pipette and positive displacement pipette tips. Pushing the pipette to the bottom position will show a metal claw. This claw holds onto the plastic piston inside the pipette tip. Firmly push down the pipette until you feel the tip lock into place. Next, label an empty 15 ml falcon tube with sputum and the sample number. Aspirate 100 microliters of selected sputum into the pipette. Ensure that no bubbles are selected. Add selected sputum in 100 microliter aliquots to the labelled 15 ml falcon tube. Then record the volume of selected sputum added to the sputum processing worksheet. Preparing smear slides. In the event where there is less than 100 microliters of total selected sputum, You will need to make sputum smear slides as you cannot continue the sputum processing protocol. First, select a single sputum plug from the selected sputum. Then dispense the sputum plug onto a clean microscope slide. Using a second clean microscope slide, smear the sputum plug across both slides. Sputum cell smears should be visible on both microscope slides. Smear slides will need to be air dried for a minimum of 1 hour before staining. Sputum and sputolysin incubation. Discard the remaining sputum sample and petri dish. To the selected sputum, add 4 times the volume of diluted DTT. In this case, 400 microliters of diluted DTT is added to 100 microliters of selected sputum. Carefully mix sputum and DTT with a pipette or a pasta pipette, depending on sample viscosity. This will initiate sputum homogenization. Place sputum and DTT tube onto a tube mixer for an initial 30 minutes. After 30 minutes, check to see if sputum sample has homogenized. If cells are not homogenized, as shown here, very quickly vortex the sample and recheck for homogenization. Again, the cells are still not homogenized, therefore mix for an additional 15 minutes on the tube mixer. As shown here, the cells have now homogenized. Sputum must be mixed for a minimum of 30 minutes and up to a maximum of 1 hour to ensure homogenization. Filtering of sputum. Sputum filter assembly. The sputum filter has 4 parts and should be made when the sputum and DTT are incubating. To assemble the sputum filter, first place filter paper onto the bottom part of the sputum filter device and align with your fingers. Next, place the O-ring onto filter paper and again align each component. Next, gently screw the top of the filter onto the bottom part. When finished, check O-ring is in correct position. Pre-wet the filter with PBS. For pre-wetting the filter with PBS, you will need the items shown. First, remove a pasta pipette from the wrapping. Next, aspirate approximately 1 ml of PBS into the pasta pipette, then dispense the PBS through the filter until you see the droplets coming out the bottom of the filter. Sputum filtration. To filter the sputum sample, you will need the items shown. Initially add PBS to sputum to inactivate the DTT. The PBS volume will also be 4 times the original volume of sputum. In this case, it will be 400 microliters of PBS added to the sputum sample. Unwrap the 10 ml syringe and the mixing cannula and attach them firmly together. Partly aspirate the sputum sample into the mixing cannula, and using the syringe with mixing cannula, gently mix the sputum sample. Then draw the entire sputum sample into the syringe, ensuring none is left in the mixing cannula. Place the mixing cannula back into its wrapper for later use. Next, attach the pre-wet filter assembly to the syringe. Now, slowly filter the sputum through the filtering device. Sputum is filtered 1 drop at a time. Now, take apart the filtering device and use the mixing cannula to lift the filter paper up for examination. Here, you are to check the filter paper for mucus and cell debris that may have been left behind during the sputum filtration. Add these details to the sputum processing worksheet. Sputum total cell count. To perform the total sputum cell counting, you will need the items shown. Label an empty 1.5 ml tube with TCC for total cell count. Add 20 microliters of trypan blue to the 1.5 ml tube. Using a new pipette tip, mix the sputum sample. Then take 20 microliters of sputum and add to the 1.5 ml tube. Mix trypan blue and sputum together with the pipette, then aspirate 20 microliters of this solution. Loading the hemocytometer. Slowly dispense the sputum and trypan blue mixture onto the hemocytometer. Capillary action will spread the solution evenly throughout the hemocytometer. Sputum cell counting. The hemocytometer will have grids marked out. These can be visually seen without the microscope. This is the section you should focus on when using the microscope. To perform a total cell count, only the cells within the blue areas shown are to be counted. Cells should be counted with the assistance of a cell counter, such as the one shown here. Place the loaded hemocytometer onto the microscope stage. Focus on the hemocytometer grid by ensuring the microscope light is passing through the grid. Count the cells using a 40 times objective. These images show typical viable and dead cells that would be seen when using the microscope. Dead cells are partially or completely filled with blue, while viable cells are completely free from blue staining. Both dead and viable cells should still look cellular. They should be roughly circular, have a grainy cytoplasm, and the nucleus should still be seen. Red blood cells are perfectly circular, have no nucleus, are opaque, and have a faint red color. Do not count red blood cells. Squamous cells are much bigger than other cells and have large nuclei. Do not count squamous cells either. Add total cell count result to the sputum processing worksheet. We will now discuss the sputum total cell counting. First, add together the number of viable and the number of dead cells. Next, to get the total cell count, divide the total number of cells, in this case 30, by the number of quadrants counted, which will always be 2, and times this by 0.02 mils to reflect the 20 microliters of trypan blue and cell suspension added to the hemocytometer. To calculate the viability, divide the number of viable cells by the total number of cells counted, and times this by 100 to get a percentage. To calculate the volume of PBS that is required to resuspend the cell pellet into a 1 by 10 to the 6 cells per mil cell suspension, you will need to times the total cell count by the total sputum sample volume. On the sputum processing worksheet, you are also required to multiply the total cell count by 9. This estimates the number of cells per mil of raw sputum in the airway. Sputum sample centrifugation. This image shows a typical centrifuge. Take the remaining sputum sample, load the sputum tube into the centrifuge, then load a balance tube that is filled to the same level with water. Next, close the lid. Set the centrifuge to a speed of 400 G for 10 minutes, and if possible set the temperature to 4 degrees. After 10 minutes, open the centrifuge and remove the balance tube filled with water and the sputum sample tube. Sputum supernatant storage. To store sputum supernatant, you will need the items shown. During the 10-minute centrifuge spin, tubes have been labelled with patient identification number and sputum supernatant symbol S/N. Add 250 microliters of sputum supernatant into each 1.5 ml microcentrifuge tube. If the volume of supernatant is large, aliquots of 500 microliters or 1 ml can be stored. When aliquoting sputum supernatant, do not disturb the cell pellet. When finished aliquoting sputum supernatant, close the 1.5 ml microcentrifuge tube lids and store the sputum supernatant at -80 degrees Celsius. Record the number and volume of sputum supernatant tubes stored. Preparing sputum cell suspension. For this step, you will need the items shown. Add the calculated volume of PBS to the sputum cell pellet. This volume was calculated during the total cell count calculations. Using the pipette, reconstitute the sputum cell pellet. Sputum microscope slide preparation. To prepare sputum slides, you will need the items shown. Place slide face up into the metal clip. Then sit filter card on top, followed by the cytospin bucket. Lock everything into place with the clip. Ensure that you can see through the hole in the cytospin adapter and that everything is aligned. Again, place slide face up into the metal clip. Sit filter card on top, followed by the cytospin bucket. Lock everything into place with the clip, and then ensure that you can look through the hole in the cytospin adapter and that everything is aligned. Open the cytospin, then pull out the rotor assembly and lift the lid by pulling the silver tab on top. Load the assembled cytospin components into the cytospin rotor. Insert them on an angle with the top of the bucket facing towards the center of the rotor. Add 70 microliters of sputum cell suspension into each cytospin bucket. Place the lid back on the cytospin rotor. Lift the silver button and then push the silver button back down to lock into place. Place the rotor back into the cytospin. The speed is 500 RPM for 5 minutes with maximum acceleration. After 5 minutes, open the lid and remove the cytospin rotor. Remove each assembled cytospin component. Gently unclip the cytospin holders so that the slides do not break. Lift the filter card off directly so you do not smear the cell pellet on the slide. The cytospin buckets need to be washed in pyron egg, and the filter cards and slides are left to dry for 24 hours. If you have followed the procedure correctly, the sputum cell pellet should be visible on the slide. Record the number of slides you have made on the sputum processing worksheet. Microscope slide staining with May-Grunwald Giemsa. Half fill 3 Copland jars with methanol, May-Grunwald and Giemsa reagents, and completely fill 2 Copland jars with water. Take your dried microscope slide and place into the methanol for 10 minutes. After 10 minutes, take the microscope slide and shake off methanol droplets, and then place the slide directly into the May-Grunwald Copland jar for an additional 10 minutes. Shake off May-Grunwald droplets and then place the slide directly into the water Copland jar. Take the Copland jar to the sink. Have a slow and steady stream of water running from the tap. To wash the slide, fill and empty the Copland jar with fresh, slow running water. Hold onto the slide with your finger while emptying the Copland jar. Rinse the slide for a total of 3 times. Shake off water droplets and then place the slide into the Giemsa Copland jar for 5 minutes. Shake off the Giemsa droplets and place into the water Copland jar for the final wash. Take the Copland jar to the sink and again have a slow, steady stream of water running from the tap. And to wash the slide, fill and empty the Copland jar with water, holding onto the slide with your finger when emptying the Copland jar. Repeat for a total of 3 rinses. Next, air dry the microscope slide with the stained cell pellet facing up. Dry for a minimum of 60 minutes. When the staining process is complete, a stained cell pellet should be visible on the slide. The chromatrope 2R or C2R staining method is similar to the MGG staining procedure. The only differences are the reagents and the staining times. Fixing stained sputum slides. To fix the slides, you will need the following items. Place 3 drops of mounting medium onto the cover slip. If you are using a smaller cover slip, use only 1 drop. Press the slide pellet side down onto the cover slip. This removes excess mounting medium and bubbles. Flip the slide and the joined cover slip over and allow to air dry for a minimum of 60 minutes before counting. Stained sputum slide examples.
We will now look at good and poor sputum samples under the microscope. Here is a sample with no salivary contamination. The background is clear and only lower respiratory cells are present. This sample has only a small amount of salivary contamination. Squamous cells and contaminating bacteria now appear on the left side of the screen. In a poor sputum sample, there are excessive amounts of salivary cells which occupy all of the background, and lower respiratory cells are not seen.
For further information on identifying sputum cells and sputum cell counting, refer to the standard operating procedures for counting MGG and C2R slides. Summary of sputum processing. This flowchart details the entire sputum processing procedure in order.